Hello and welcome back to Principles of Organismal Biology. Today we're going to start learning about plants. Yes, that group that it's in Archaeoplastida. And we're going to be covering some of the most important or most exciting organisms on Earth. Okay, let's start. So today we're going to learn about Archaeoplastida. That's one of the 4 supergroups in Eukarya. And we're going to distinguish between green algae and land plants. Importantly, we're going to be looking at the particular features that make land plants, and some of the main adaptations that they have to overcome in order to move to land. We're going to discuss also if these plants form a monophyletic group or not. And in particularly today, we're going to learn about bryophytes. And that's a lineage, an early evolving lineage in land plants. So we're going to look at this, and just to put you in the context of where we are right now. So we are looking at eukaryotes, right? So we have talked that we have 3 domains. Eukarya is one of them. And then if we zoom in, last class we covered Uniconta. I want to emphasize that because Uniconta is one of the supergroups that includes fungi and animals. Last class we covered fungi. Today we're moving to learn Archaeoplastida. And one of the questions that I want to ask you is, how did plants change the world? And I'm talking about plants as land plants, and also they are called embryophytes, or plants that have an embryo. So we are looking at this group of Archaeoplastida right here. And if we look at this phylogeny, you could look at the ancestor, most common recent ancestor of plants and caryophytes. So right here we have an ancestor, and this means that caryophytes are the sister groups of plants. Plants are multicellular photosynthetic eukaryotes, and they are going to have cell walls, and in comparison to, for example, fungi, plants have a cell wall made out of cellulose, right? Fungi have cell walls made of chitin. So it's a different type of polysaccharide that plants use to have this cell wall. Since the colonization of land, plants have diversified in thousands and thousands of species. Today, we have around 3,000 living species, and we keep adding more and more species into our list. So some of them have returned to the aquatic environments. Most of them live inland, and some of the ones that have returned to the aquatic environments are, for example, seagrasses. Algae are not included in the plant kingdom. So algae are considered in the kingdom system. They were considered algae, but they are photosynthetic protists, right? So they photosynthesize, and now we understand that the chloroplast is a result of this endosymbiotic relationship of a heterotrophic eukaryotic cell and a cyanobacteria. And that chloroplast is present in plants, land plants, but also algae. Benefits that plants have to us is that they provide us with food sources, but also oxygen. So if you take a deep breath right now, a big percentage of this oxygen comes from these plants. This is another phylogeny for us to understand the evolution of plants. And here we have all of the Archaeoplastida. Archaeoplastida has the groups of red algae, green algae, and land plants. And in here we have different lineages as well, included in this green algae definition. So if we look at this ancestor right here, these lineages evolved from that common ancestor, and that led to the evolution of embryophytes and caryophytes. So this caryophyte, therefore, is the closest relative to all land plants. That means green algae are the most similar to plants. What's the evidence to suggest that? So obviously we have a lot of genetic data now, but other than that, we have other type of evidence. So we have some features that Bryophytes and plants share, and they are not present in other algae, for example. That is the presence of rings of cellulose. So cellulose is going to be this outside layer that plants have outside of the membrane, the plasma membrane. In here you could see this membrane. It's a phospholipid membrane that we can find in plants, that we also have, but there is a complex of cellulose synthase proteins or subunits, 6 subunits that assemble, and they are going to be synthesizing or helping to synthesize this glucan change that makes the cellulose fiber. This cellulose is going to be very intricate built, and other molecules are also going to be enhancing or supporting this cell wall. So that's the first thing that Bryophytes and plants share, the rings of cellulose, right? So these 6 subunits that form this ring, that's important. In here we can see microphotography that has these subunits, and from here the cellulose fibers will be coming, going outside, towards the outside of the cell. I want you to think about what you have learned in the class and match the figure with the content in the cell wall. And I want to match the figure of cyanobacteria. In other words, what's the cell wall of cyanobacteria made out of? A, cellulose, B, chitin, C, external layer of peptidoglycans, or the internal layer of peptidoglycans. Okay, so if you remember, we say that fungi have the cell wall made out of chitin. We just covered these. Plants have the cell wall made out of cellulose, mainly. In gram-negative bacteria, we have actually an internal layer of peptidoglycans, so D is the right answer, because the peptidoglycan is in between these 2 membranes of lipids, the plasma membranes. And in the gram-positive bacteria, such as, for example, E. coli, we will have this peptidoglycan layer in the outside, okay? So D is the right answer for cyanobacteria. The second feature that plants have, or that they share with Bryophytes, is the structure of flagellated sperm. In here, we can see some plants that have flagellated sperms. A lot of plants, or most of the plants that are on Earth right now, lack flagella, and we're going to learn that later on when we talk more about pollen. But some plants, some of the early evolving lineages have flagellated sperms, and that's something that they share with Bryophytes. So that's the second feature. And the third feature is the presence of phragmoplast. Now, I want you to think about what happens in mitosis. In mitosis, we're going to have the division of the cells, right? So in telophase, we are going to have the nuclei migrating to both sides. This is one nuclei, or one nucleus, and the other nucleus is right here. And then the first thing that it needs to happen is that we have to form part of the cell wall, because the cell wall is going to be, the thing that also divides these 2 cells. And we are also going to be dividing the plasma membrane. So that's what we refer to cytokinesis, right? Karyokinesis is the division of the two nuclei, but cytokinesis is the division of the plasma membrane. So the phragmoplast is a type of a scaffold that helps in these early stages of the cytokinesis, the division of the plasma, the cytoplasm in these two daughter cells. And this scaffold is made out of microtubules. So that property is shared between land plants and Bryophytes. So this is the three evidence on top of the genetic information that we have to support that Bryophytes is the closest living relative to plants. One of them is of the living Bryophytes are Sygnema, or members in the Sygnema genus, and another genus is Coleochaete. And this is one of the algae. This is how they look. So they are multicellular organisms that photosynthesize, right? And they have cell walls. So that's what I was telling you. Depending on the definition, you can call plants to something different versus another definition. So let's look into what makes land plants or embryophytes. In order to do that, we need to understand some of the adaptations and innovations that plants overcome in order to colonize Earth, or some of the challenges that they overcome. And I want you to look at this figure right here. We have an algae, and this, or alga is singular, and this alga has a lot of water, right? It has water, and it absorbs the water directly, but it absorbs the CO2 that is contained in the water, and minerals from the water as well. So it is very different. It's a different game, or different strategy, or multiple strategies that organisms living in aquatic environments have compared to the ones that have to live in land. Let's look at the conditions that land plants have. In here, this plant has more access to CO2. There is more CO2. There is more light, and you know that these two groups photosynthesize, so therefore they need light to photosynthesize, and there is more light in here versus in the aquatic environment. What's one of the challenges, or one of the challenges that plants have is desiccation, right? So now we don't have that much availability of water. So the water actually is coming or associated to the soil, so the substrates in which plants grow, and the nutrients are also in this soil. So plants have evolved this root system to anchor, but also to absorb water and the minerals from the soil. So these are some of the benefits, the benefits that plants have colonizing Earth. They also didn't have to compete with other individuals in the sea or in the aquatic environments. Now they have all this land to explore and to reproduce there, but they had to overcome the challenges. Some of these adaptations that they have evolved to overcome the challenge of desiccation is the spore pollenin. Spore pollenin is going to be a layer, it's a lipidic layer that gets deposited in some spores. And these spores, for example, here, are covered with spore pollenin. Spore pollenin is also found in some, in pollen, for example, but it is present in the spore walls. I want you to take a moment to read this question and think about it. But then some lineages of plants living in humid environments have flagellated sperm that swim to fertilize the female gamete. As the environment turned drier, a group of them evolved to be unflagellated and develop thicker walls with spore pollenin. and they left more progeny than the ancestral flagellated version. Based on this description, only you can conclude that this occurred by A, the fact that the genetic code is universal, B, gene flow, C, horizontal gene transfer, D, mutation and natural selection, E, hybridization. Well, hopefully that by now you realize that every single new variant appears by mutation, and the natural selection only fixes that variation or that variant in the population or purchase it, right, from the population. So B is the right answer, mutation and natural selection. So to recap this idea that of the adaptations that plants had, so once that they move to land, they have more sunlight, they have a lot more CO2, and they have a nutrient-rich soil. And they had to deal with lack of water and lack of structural support. And if you think about that, think about an algae, taking an algae, hopefully you have hold an algae in the past, and if you hold it and try to put it to a stand, it will fall. So they lack this structural support, and they will just fall because of gravity. So those are some of the key features that you need to remember associated to the adaptations that plants evolve to be successful on Earth. And those adaptations are the ones responsible for plant diversification. As I said, we have now more than 300,000 species of plants. Now, land plants, as I said, if we only look at land plants, we can define them as plants that have embryos. And embryos are going to be this multicellular structure that is the result of a zygote, right? So it's diploid in most of the cases. And they are going to be retained, like in this case, and we are going to have a placenta. So maternal tissue that is fitting this embryo. Now, this is again the phylogeny that I mentioned before that includes all of the Archaeoplastida, or all of the members in Archaeoplastida. This, at the base in this ancestor, the ancestral aga, is the one that had the primary endosymbiosis right here. And also all of these guys, all of these members of Archaeoplastida are going to have the presence of chlorophyll A and B. And later on, or maybe you already know, chlorophyll A and B are some of the pigments, the chlorophyll molecule is necessary to perform photosynthesis. Now, if we look at this right here, these are the three features that we talk about it. The rings of cellulose that Bryophytes and land plants have, the presence of some flagellated sperms, and the phragmoblast. Okay. Now, if we look at the embryophytes only and look at the synapomorphies, so those features that we are only going to be or find associated to embryophytes, we're going to have, the presence of embryos. We're also going to have the presence of cuticle. And cuticle is this layer. So have you ever watered a leaf of oak, for example, or other leaves? If you haven't done it, just go outside right now and pour a little bit of water on the top of some leaves. What happens? Observe what it happens. And hopefully I'm not wrong, but what usually happens, or what it happens, is that those drops of water they run off. So plants do not absorb the water from the leaves. They absorb it through the roots, the root system. And they cannot absorb it because they have this cuticle layer, this lipidic layer that protects them from the sun. So that's another adaptation that land plants have that make them successful into moving into land. Others are alternation of generations, multicellular dependent embryos, the wall spores produced in sporangia, so more protected spores, multicellular gametangia that are also protecting the gametes, and the apical meristems. So let's take a look to these 5 in detail for you to understand what we are talking about. So alternation of generations. We talk about alternation of generations when we introduce brown algae, I believe, and we define that in alternation of generations, you require 2 multicellular generations. In this life cycle, this is the life cycle of a fern, we have 1 multicellular organism in here, and this is diploid, and another multicellular organism that is haploid. So that's the alternation of generations. So we have 2 multicellular generations, and that is key. That's going to be found in algae and in plants, in some algae and in all plants. The gametophyte right here, this word is the generation that is haploid. And the sporophyte is going to be diploid. Now, it's very easy. Well, I know these are new terms for you, for most of you, but it's really, really not that complicated. You just have to look at the root of the word. Gameto reminds you, hopefully, of gametes. And phyte refers to the plant. So the plant or the organism producing the gametes, that's the gametophyte. Now, the sporophyte, it's the organism that is going to be producing the sporophytes. So very simple. And the other key element that you need to remember is that the spores are going to be the result of meiosis, and that the spores develop into gametophytes. So in plants, the spores develop into gametophytes. Do humans have alternation of generation? So we have meiosis, right? We form sperms and we form ovules, but do we have a multicellular haploid stage? What do you think? Okay, to exemplify that we don't have alternation of generations, I'm going to bring one of my favorite actors. Right, I love Brad Pitt. This is Brad Pitt, and what he does is that, you know, some cells go through meiosis, and they just imagine, this is imagine how alternation of generations will look in humans if we do have alternation of generations. That means that now Brad Pitt will be producing spores, because that's what he does. He produces spores, and then these spores develop into a haploid multicellular organism, and that's the version that I came up with. And then that organism will form or will produce, through mitosis, will produce sperms, right? And then that sperm will fuse with an ovule, result of also an alternation of generation from, I don't know, a woman, whatever you want. And then that will fertilize and produce a zygote. And then that will be a baby girl, baby boy, whatever, and we'll close the cycle. You see, we don't have that. We don't have this in humans. So in humans, we directly, from meiosis, we skip straight into the sperms. So we don't have this multicellular organism that is haploid. I hope that helps for you. I know it's a little bit silly, but it really helps to clarify why humans do not have alternation of generations. Okay, so alternation of generations is present in land plants, not in caryophytes. The second derived trait of plants is this multicellular dependent embryo that I briefly talk. So we have these cells, this group of cells that are the embryo. They are diploid, so they are the result of fertilization. And we can see also that some of the maternal tissue surrounding this embryo is exchanging nutrients, or it's providing the nutrients to the embryo. And you can see that because we have these wall ingrowths that facilitate the connection of the plasma membrane between the maternal tissue and the embryo. The third feature that is present in land plants only is the wall spores, but not only that the spores have this wall that I mentioned before with the sporopollenin, but also that they are being produced inside of these sporangia. In here you can see a beautiful landscape of mosses covering a decaying forest, or a forest that actually is very healthy, and it has a lot of nutrients. All of these green little plants, most of them are mosses. In mosses, we're going to learn in a second that they are part of the bryophytes. Bryophytes is an artificial group of land plants. But these mosses, if we zoom in into this landscape, like right here, we'll see that some of these mosses look like that. And the sporophyte, the one that produces the spores, is this structure. And the one that looks more grassy is actually the gametophyte. The one at the bottom is the gametophyte. Well, now in here, in this sporophyte, we have this structure at the top that is called sporangia in plural, or sporangium, it's singular. So here we have one sporangium full with the spores. That's the third feature that is only present in plants. The other feature, the fourth, is multicellular gametangia. So remember, the gametophyte is the structure or the organism that is haploid, that is going to produce the gametes. But then the gametangia is actually a structure in the gametophyte. So the gametophyte is all of these structure, and the gametangia are going to be right here. And the gametangia can be an archegonia or an antheridia. The archegonia is going to be producing the eggs, as you can see right here. So these are structures, multicellular structures, the gametangia, that are producing the eggs in yellow, or in this case, the antheridia is going to be producing the sperm. And the sperm is going to be swimming in these environments. So they are very humid environments. They need water for the sperm to swim, right? So this is one of the bryophytes, but it's not a moss. It's liverwort. This one in particular belongs to the genus Marchantia or Marchantia. The fifth feature that land plants have is apical meristems. And the apical meristem is a structure that has rapid divisions, mitotic divisions, that are either at the tips of the shoots or the tips of the roots. So you can find them there, and they are going to be protected by some surrounding tissue in the case of the roots, or by some type of baby leaves, developing leaves, that protect the apical meristem of the shoot. Okay, so that's the fifth. Now, let's talk about the origin and the diversification of land plants. They appear in the fossil record for at least 470 million years. So since we know that land plants at least evolved 470 million years. And that's because we have found in the fossil record, we have found some fossilized pollen. Sorry, I'll take it back. We have found some spores, fossilized spores. Then the composition, the molecular composition of these spores is similar to the composition of members in bryophytes. So that tells us that these species are most likely, so not only morphologically, but also at the chemical composition, it's most likely a member of the land plant group. Other fossil that has been found that it's a fossilized sporophyte. And that's the tissue that is going to be producing the spores, right? So that has been dated at least 450 million years old. This is a table that summarizes the different groups that we have in land plants. And in here we have the non-vascular plants, bryophytes. So some of the ones that I was presenting already, mosses, liverworts, belong to this group bryophytes. You can see that liverworts have around 9,000 species. The most diverse bryophyte is the mosses. Mosses have around 15,000 species. And the less diverse is a group called the hornworts. And then we have vascular plants. In next class, we're going to talk about what makes vascular plants vascular plants. But for today, just look at the diversification in these groups and look at the highest diverse or the most diverse group, which is the angiosperms, the flowering plants. Today, we're going to focus now into non-vascular plants, this group that we call bryophytes. In this group of plants, gametophytes are going to be the dominant phase. So that means that they live the longer in comparison to sporophytes. Based on this phylogeny, bryophytes are monophyletic, yes or no, or true or false? Okay, hopefully you answer false, because the ancestor of all bryophytes, so let's ask the question, where is the most common recent ancestor of all bryophytes? So liverworts, mosses, and hornworts are bryophytes. So if we look back, so the common recent ancestor, that means the ancestor that unites all of them, will be in number one, right here. Our definition of monophyletic is that a monophyletic group has the ancestor with all of the descendants. And in this case, we have the ancestor with some but not all of the descendants. And so that means that this bryophyte group is paraphyletic. So bryophytes is an artificial group, and it was made based on the lack of vascular tissue. So as I mentioned before, they don't have vascular tissue, so they are not woody plants. They are small, right? They are very small in comparison to any other plants in other groups. They tend to be very small. So we have three groups: liverworts, mosses, and hornworts. And these three groups evolved early in the history of land plants. So we refer to them as the earliest lineages. This is the life cycle of a moss. So let's take a look into that. And just remember that the gametophyte is the dominant phase. So let's start in this structure with the production of the spores. So spore is going to be haploid. Remember, for consistency, everything in turquoise is haploid. And these spores are going to be developing in a protonemata. These protonemata are a group of cells that they are a series of cells, but it's just one cell in thickness. And these protonemata later will produce a bud, and we call them bud because they are not exactly compared to the buds in other plants, in vascular plants, for example. But they produce a type of a bud, and this bud will develop or differentiate into the gametophyte. So this is a male gametophyte, and here we have a female gametophyte. And in this, just remember, the gametophyte is the dominant, and it's haploid. So this structure, this organism, now is going to be producing the gametes. And these gametes then are going to be produced through mitosis. So the division of certain cells produce the eggs, haploid eggs, and in here in the antheridia, we're going to have the formation of sperms, haploid sperms, of course. And when there is water around, or during the rainy season, this will happen. The sperms will get released and they will travel in the water in this film around the plants or around the soil, and then they will climb and reach the swim, I meant to say swim. They will swim up to reach the eggs in the archegonia of the female gametophytes. So fertilization will occur, and what's the result of fertilization? Well, it's a zygote. So we reestablish the diploid condition right here in the archegonium. In this, we're going to have this zygote in this archegonium, and this zygote is going to now develop into an embryo. So it's going through mitosis. So this is diploid, and this diploid embryo develops into the young sporophyte. So that's diploid still, but notice that this sporophyte gets retained into this gametophyte or gametophytic tissue. So everything in green right here is haploid. Now this sporophyte keeps developing and produces these structures. So the foot is the one that anchors it to the gametophyte. Then the seta is this kind of stalk, and the capsule, which is what we referred to the sporangium before, is the one that produces all of the spores. But the spores, remember, are going to be produced through meiosis. The spores are going to be produced through meiosis. So now think about this. Bryophytes are very, very small, very short. Some of them can reach higher heights, but most of them are very, very, very short. So they form mats on the ground. And the reason why they don't grow that much is because they are very thin, but also because they lack vascular tissue. So the rhizoids that they develop, they're called rhizoids. They have a similar function as roots. So roots in other plants, not in bryophytes. So bryophytes do not have roots. They do have rhizoids. So roots and rhizoids anchor these plants, but rhizoids do not have vascular tissue. So they evolve similarly, they look similar by convergence, but they are not the same structure. This is what happened in the sexual life cycle. However, bryophytes can also reproduce asexually through brood bodies. So this is one brood body that this plant, the gametophyte, will produce and drop, and this will anchor into the soil and will become a new plant. So this is another way that these bryophytes have to reproduce by asexually, so they will be clones. What is important for you to remember is that the sporophytes depend all the time in the gametophyte. We'll learn later on that in other plants, sporophytes and gametophytes can live independently, but in this group, they cannot. So sporophytes depend on the gametophyte, and they are the smallest and the simplest sporophytes of all of the plants on Earth. The sporophyte is going to consist, as I mentioned, the foot, the seta, and the capsule or the sporangium. And the peristome is this structure that opens to release the spores. And this is really clever. This is a structure that opens with certain properties, like wind. If there is wind, they will open and they will disperse the spores, but if not, they will get closed. And also with other environmental features like water, humidity, they respond to all of these conditions. And here we see, now, one property that plants have is the presence of stomata. And stomata are going to be pores in plants that allow for gas exchange. Hornworts and mosses have stomata. They are going to have sporophytes that have stomata, but liverworts do not have stomata. Okay, that's something that these guys lack. This is liverwort right here, and this is the sporophyte of the liverwort. This is structure do not have stomata. So if you are not familiar with the stomata, I'm going to give you a quick introduction. These are the pores in plants. In most of the plants that you are familiar with, they look like that. They will have leaves. So these are vascular plants, not the bryophytes that we were just discussing, but they have some holes. They are called stoma, and the stomata it's referred to this hole, but also these guard cells. So these cells that are surrounding this hole, and these guard cells respond to difference in solutes. So they are going to change based on the osmotic pressure, and that's also triggered by some hormones. That's a very ingenious way that plants have to regulate the aperture of this stomata, because when the stomata opens, the plant exchange gases, so it will take CO2 from the atmosphere, but it will also release oxygen, right? So that's very important. But the other thing that happens is that plants lose water in the form of vapor through the stomata. So they really need to regulate if they want to keep water, or if they need to open these pores to exchange gases and photosynthesize, because they need the CO2 to fix that carbon into some organic molecules such as glucose. Okay, let's explore this diversity. So in hornworts, this is the phylum Anthocerophyta. And phylum is usually referred to animals. In plants, they are called divisions, but your book uses phylum, so I'm going to be using it as well. So hornworts belong to this phylum Anthocerophyta, and that refers to this type of arrows, or that the sporophyte has the shape of this stalk. The gametophyte will be at the bottom in this group. In the case of mosses, we have all of this is the gametophyte, all of the greener portion, the more hairy, or the hairier part, the one that looks grassy, is the gametophyte. And in here you can easily see these different parts of the sporophyte. That's the seta and the capsule, right? And in this capsule is where we're going to have the spores being produced and released. So again, these mosses can form large mats, and they're going to be very important in some ecological processes I'm going to discuss later. Some of them are associated, some of these mosses can be associated with nitrogen-fixing cyanobacteria or other type of microorganisms that help fixing nitrogen. If you remember, I have mentioned that 72% of the content in atmosphere is nitrogen. So we have 72% of the air is nitrogen, but that's a type of nitrogen, and two, that is not easily accessible by plants. So some organisms in the soil can convert that nitrogen, this molecular nitrogen, in other forms, in other molecules that now the plants can acquire. And that's what it has been shown in this figure. Some researchers measure how much nitrogen is lost in absence of mosses. So with moss, if we have a land cover with mosses, we will have the loss of nitrogen will be very minimal. And however, if we remove the mosses, the soil will lose the nitrogen more. And that's because then now we don't have this microhabitat in which these cyanobacteria or nitrogen-fixing cyanobacteria thrive, and we will be losing that as well. Another important aspect associated with mosses is the formation of peat, and this peat has been formed by a sphagnum. Sphagnum is a genus in these mosses, in these bryophytes, and they form layers and layers of mosses that they die, but they don't decay. And these have a lot of carbon accumulated. So look at the top, now we have mainly grasses, but in the past, these had mosses, a layer of mosses that died, and on top of them were growing new mosses and new mosses. And so we accumulate a lot of these organic materia in this pet. And the reason why they don't decompose is because there is low temperature in some parts of the earth in which these mosses grow. We will have low temperature, low pH, and the oxygen will be also very low. So all of these conditions inhibit the reproduction of microorganisms that can decompose this moss. So that creates this particular environment in which carbon can be accumulated. And it's very important to consider that this process has store carbon. If we consume this pet as fuel, as it can be used as fuel, but also if we keep buying it for our gardens, they now can decompose, and all of this carbon gets released to the atmosphere, and that can also increase the temperature in our planet. So it's very important that we preserve these peatlands as they are. The other curious and very surprising phenomenon that happens around the presence of these mosses is that these conditions of low temperature, low pH, and low levels of oxygen can preserve or have preserved humans. So this is a Toulon man that was found in some of these pet areas, and you can see all of the facial expressions on these men. It's incredibly well preserved. So it's a type of subfossilization that these lands have helped to preserve. So peatlands cover 3% of Earth's land surface and contain around 30% of the world's soil carbon. So just think about it. If we burn these peat areas, we will be releasing 30% of this carbon. So we need to stop that. Before I stop this class, I want to give you a couple of tips to prepare for your exam. And what I would like you to remember is that the life cycles are going to be critical for you to remember, to understand the differences. In here we have the life cycle of animals, plants and some algae, and then fungi and some protists. I want to ask you, what of these life cycles have alternation of generations? Okay, so A, animals, B, plants and some algae, or C, most fungi and some protists. The right answer is B, plants and some algae have alternation of generations. So in here, we have this sporophyte that is diploid, right? And here the gametophyte that is haploid. Both of them are going to be multicellular. That's our definition of alternation of generations. I have mentioned when I presented the life cycle with Brad Pitt, that was to exemplify that humans and animals do not have alternation of generations. So the result of meiosis is going to be the gametes. Whereas in this case, the result of, in the case of plants and some algae, the result of meiosis is going to be a gametophyte. And then this gametophyte is going to produce gametes. So we have a multicellular stage that is haploid. We have also a multicellular stage that is haploid in fungi and other protists, but the difference is that the zygote it's the only stage in which we have a diploid condition. So we don't have a multicellular diploid condition in fungi or protists. Hopefully this helps you to summarize the main differences in these life cycles that we have presented. The other tip that I want to give you is to understand the different classifications that we have. So before we have the 5 kingdoms that classify everything, and now we have the 3 domains. So we have gone through all of these aspects, but I wanted to summarize for you to help you going through this process. So the 5 kingdoms will have Monera, Protista, Plantae, Fungi, and Animalia. So those were the 5 kingdoms that we were using before. Now, with the advancement in technological aspects, in particular with massive sequencing, we now realize that actually members of archaea or archaea and eukarya are more closely related than archaea and bacteria. And Monera, then therefore this kingdom, this artificial group that also Monera is equals to prokaryotes, was split in these 2 domains, the bacteria domain and the archaea domain, right? Now, the other 4 kingdoms, Protista, Plantae, Fungi, and Animalia, the 4 of them fall into this eukarya domain, okay? Now, also associated to all of these sequencing results or the data that we have generated recently, we know that the eukarya, that these kingdoms are artificial. Some of them are artificial groups. In particular, we have looked at the Protista and how they are really all over the place. So that is what has led to this reclassification and into the 4 supergroups of the domain eukarya. And these are the 4 supergroups: Excavata, Sar, Archaeoplastida, and Uniconta. For you to understand what this difference, I want to ask you where fungi and animalia, these 2 kingdoms, fall into this new classification using the supergroups, these 4 supergroups. Okay, Uniconta includes both animalia and fungi along other protists. Hopefully that helps you to understand how this reclassification has worked. And also, we have discussed extensively the evidence that we have to reclassify these species in this new system of the 4 supergroups. Thanks for joining me today, and next class we'll be continuing learning about plants. If you have any question, please email me, and if not, just come to my office hours. Thank you very much. No, please come to my office hours, please, and if you cannot make it, email me. Have a good day, and see you next time.